Imagine this. You walk into your doctor's office. You've been exhausted for months. You can't sleep. Your libido is non-existent. Your brain feels like it's wrapped in wet cotton. You gain weight doing all the same things you used to do to stay lean. You're irritable. You feel old, but you're not that old. Your doctor runs blood work. A few days later, you get the call, or worse, a portal notification with no context, and it says, your labs are normal, everything's fine, but you don't feel fine. You feel the opposite of fine. Here's what your doctor may not have told you. Normal does not mean optimal, and today we're going to pull back the curtain on exactly how normal gets defined in the first place and why people get ignored, and why those definitions may be completely failing you. So let's start this conversation with a lesson in statistics. Basically, everything in conventional medicine flows through one concept, the bell curve. Picture a graph that on the left side, low values, and on the right side are high values. In the middle is a hump, the shape of a bell. The vast majority of data points cluster in the center, and that curve tapers off at both ends. This is called normal distribution, or a Gaussian distribution. It's the mathematical pattern that appears when you measure almost any biological trait across a large population, whether that's height, blood pressure, cholesterol, and yes, even hormones. When the laboratory wants to establish a normal range for, say, testosterone or TSH, they test a large group of people. They plot all those values on a bell curve, and they draw lines at the 2.5th percentile on the left and the 97.5th percentile on the right side. That middle 95%, that is your reference range. That is what gets printed on your lab report. Now, here is the first problem with that approach, and it's a big one. Those people aren't necessarily healthy. The reference population for most labs includes whoever was tested at that lab, which means often that the people who went to see their doctor for a reason, they're already sick. Not to mention the population as a whole is a lot more unhealthy than before. The cutoffs are statistical, not physiological. They don't tell you what level is associated with feeling great, performing at your best, or having optimal health outcomes. They only tell you where that middle 95% of tested people happen to fall. The National Academy of Clinical Biochemistry has openly acknowledged that this is an issue, pointing that truly healthy individuals often have much narrower ranges than the broad population ranges suggest. But it's deeper than that, because there's a second problem, one that's been building for decades. Now think about this question. What happens to normal when the entire population shifts? If every generation is a little bit heavier, a little bit more sedentary, and a little bit more exposed to toxic chemicals like we are today, and their hormone levels are lower as a result, the bell curve doesn't stay fixed, it slides. The whole curve shifts to the left, and what used to be low becomes the average. This is not hypothetical. This is exactly what has happened with testosterone. In 2007, researcher Thomas Travison and colleagues at the New England Research Institutes published a landmark study in the Journal of Clinical Endocrinology and Metabolism. This surprised the medical community. Using the Massachusetts Male Aging Study, which tracked over 1,500 men across 3 data waves from 1987 to 2004, they found something startling. The average serum testosterone dropped from roughly 501 nanograms per deciliter at baseline to 391 nanograms per deciliter by the final follow-up. That's about a 1.2% decline per year. And critically, this decline was not age independent. That means a 60-year-old man in 1988 had a measurably higher testosterone than a 60-year-old man measured in 1995, even after controlling for age, weight, and lifestyle factors. More recent research has confirmed and extended these findings. A 2020 survey using National Health and Nutrition Examination Survey data on over 4,000 men tested from 1999 to 2016 found that mean testosterone levels continued to fall. Among adolescent young males, testosterone deficiency is now a prevalence of around 20%. 1 in 5 young men. The researchers at the MDPI published a review in 2025 noting that these declines are associated with reduced fertility, metabolic dysfunction, cardiovascular disease risk, mood disturbances, and diminished quality of life. So when your lab report says that your testosterone is 350 nanograms per deciliter and that falls within the normal range, you need to ask, normal compared to what population, measured in what year? Because the reference range itself is a moving target, one that may have drifted well below where human biology was even designed to function. The next question we really need to ask is why? Why is this even happening? The honest answer is it's multiple factors converging all at once, but research is increasingly pointing to one category of culprit that doesn't get nearly enough mainstream attention: endocrine disrupting chemicals. These are compounds that interfere with your body's hormone signaling system. They can mimic hormones, block hormone receptors, or disrupt how hormones are produced and even metabolized, and they are everywhere in modern life. Bisphenol A, or BPA, is one of the most well-studied. It's found in plastic bottles, food can linings, thermal paper receipts, and countless other everyday items that we use. Multiple studies have shown that BPA can bind to androgen and progesterone receptors, disrupting testosterone signaling even in low concentrations. Then there are phthalates. These are the chemicals that make plastics flexible. Research published in the Journal of Clinical Endocrinology and Metabolism demonstrated that phthalate exposure is inversely associated with testosterone levels across all age groups. In boys from ages 6 to 12, increased phthalate concentrations were linked to a 24 to 34% drop in testosterone. In adult women, the drop was 10 to 24%. The Endocrine Society has taken an official position on this, calling plastics-related endocrine disruptors a serious public health threat. Add this to the compounding effects of chronic sleep deprivation, which directly suppresses testosterone production, metabolic obesity because adipose tissue converts testosterone to estrogen, chronic psychological stress, which elevates cortisol levels, and cortisol is the hormone enemy of testosterone. Processed food diets, high in xenoestrogens, and sedentary lifestyles, and you have a perfect storm for hormone depletion in the population. And what's the result? A reference range that keeps getting dragged lower generation after generation while your doctor compares you to the same sinking average. Now let's zoom out for a little bit, because testosterone is just one piece of a much bigger puzzle. Your hormonal health is a symphony, and when one instrument is off, the whole orchestra sounds wrong. Testosterone. Both men and women produce it. Men produce it primarily in the testes, whereas women produce it primarily in the ovaries and the adrenal glands. In men, the symptoms of low testosterone include fatigue, low libido, erectile dysfunction, depression, poor concentration, loss of muscle mass, and increased body fat. In women, low testosterone manifests as low libido, poor motivation, difficulty building muscle, brain fog, and low energy. The critical distinction to understand is total versus free testosterone. Total testosterone measures everything in your bloodstream. Free testosterone measures what's actually available to your cells. A man can have a normal testosterone of 580 nanograms per deciliter, but if a carrier protein called SHBG or sex hormone binding globulin is elevated, his free testosterone can be functionally deficient. His cells aren't getting the signal. He feels every symptom of low T, yet his doctor looks at his total number and says he's fine. There are three forms of estrogen in women. Estradiol is the primary active form, and it governs everything from bone density and cardiovascular health to mood, memory, vaginal tissue, and skin. The conventional approach is to measure estrogen in isolation. The more sophisticated approach recognizes that estrogen must always be understood relative to progesterone, which brings us to progesterone. This is the most underappreciated hormone in women's health. Progesterone counters estrogen. It supports healthy sleep, reduces anxiety, and plays a critical role in the nervous system. It's classified actually as a neurosteroid. Progesterone is often the first hormone to decline as women enter perimenopause, sometimes as early as the mid-30s. When progesterone falls without matching fall in estrogen, you get a state called estrogen dominance. Not necessarily because estrogen is too high, but because it's unopposed. The symptoms: PMS, heavy periods, breast tenderness, anxiety, sleep disturbances, weight gain, and mood swings. Women in this state often have estrogen and progesterone values that both fall within conventional normal limits, but the ratio between them is completely off. Standard labs miss this entirely. DHEA is produced by the adrenal glands. It serves as a precursor to both estrogen and testosterone. Peak production happens in your mid-20s and declines steadily with age. Low DHEA is associated with a poor immune function, fatigue, cognitive decline, and reduced sex hormone output. It's rarely tested on a standard panel. Cortisol, which is often called the stress hormone, has a complicated relationship with all the other hormones we've discussed. When cortisol is chronically elevated due to stress, it competes directly with testosterone and suppresses progesterone production and drives a cascade of metabolic dysfunction. The body essentially says survival mode is way more important than reproduction, and cortisol wins every time. Your sex hormones lose. And finally, we cannot end this discussion about hormonal health without discussing the thyroid, because thyroid hormones regulate the metabolism of every cell in your body, and the conventional approach to thyroid testing, in my opinion, is frankly way inadequate. Most doctors run only one test, which is the TSH, or the thyroid stimulating hormone. TSH is actually a pituitary hormone and not a thyroid hormone. It is the signal your brain sends to the thyroid when it senses the thyroid output is low. A high TSH means the pituitary is shouting louder because the healthy thyroid is not responding well. A low TSH usually means either overproduction or suppression. The conventional reference range for TSH is roughly 0.5 to 4.5 micro international units per milliliter. But here's the problem. The National Academy of Clinical Biochemistry has proposed that the true normal range in healthy people is more likely to be 0.4 to 2.5. Research published in PMC supports this, showing that in healthy populations, the median TSH is around 1.5, well below the upper limit that conventional labs still accept as normal. Someone with a TSH of 4.2, technically within the normal standard ranges, may be experiencing genuine hypothyroid symptoms: crushing fatigue, unexplained weight gain, hair loss, cold intolerance, constipation, depression, brain fog, dry skin, and their doctor will just look at the 4.2 and say, it looks fine to me. But the TSH doesn't tell the whole complete story. You need to measure the free T4, which is the inactive thyroid hormone, and critically, the free T3, which is the active hormone that our cells actually use. Some people convert T4 to T3 very poorly. Their TSH looks normal, their T4 looks fine, but their T3, what their cells actually are using, is in the tank, and they feel terrible. Studies have even found an association between elevated TSH, which is within the normal range, and an elevated cortisol in otherwise healthy young men and women, suggesting that even subclinical thyroid dysfunction creates systemic hormonal ripple effects. And this brings us to the most important concept of this entire conversation: the difference between normal and optimal. Normal is where 95% of the tested population falls. Optimal is where you feel, function, and perform your best. And it's where research associates the best long-term health outcomes. These are not the same thing. When clinicians talk about optimizing hormones, they're talking about targeting the upper portion of the healthy physiologic range, not just clearing the floor of the reference range. The Endocrine Society and major urology bodies have studied that men on testosterone therapy should be maintained in the mid-hormonal range of roughly 450 to 900 nanograms per deciliter. They do not suggest just pushing testosterone levels above the floor at 300 and call it a day. For women, optimizing means looking at the full picture: estradiol, progesterone, free and total testosterones, DHEA, SHBG, cortisol, free T3, free T4, and TSH, and evaluating how they interact with each other, not just whether each individual number clears the bar. Optimization also means tracking the symptoms and not just the numbers. If a patient has a testosterone level of 400 and is symptomatic and feels dramatically better at 650, this is clinically meaningful information. The goal isn't to treat a lab value. The goal is to restore a person to how they're supposed to feel and function. And this is a paradigm shift, because conventional medicine asks, are you sick? Or hormone optimization asks, are you thriving? Are you at your best? So the next time you get lab results back and they say normal and something still feels deeply off, remember what you know. Normal is a statistical average drawn from an increasingly unhealthy population, a marker that keeps moving to the left. It's a floor, it's not a ceiling. It tells you that you're not at the absolute bottom of the curve, but it does not tell you that you're functioning at your biological potential. The questions to ask your provider are, what does optimal look like for someone my age and sex? Are you measuring free testosterone, not just total? Are you looking at T3 and not just TSH? What is my progesterone to estrogen ratio? What is my SHBG? Has anyone looked at my DHEA? Advocate for that full picture, because you deserve to feel just not sick, but you generally deserve to feel vitality and unmistakably well. Thanks for listening. We'll see you on the next episode.